All right. Oh, all right. Oh, boy, I can use one. Yeah. Okay, well, I guess our last thing in our, our last thing is we're going to take just a little look about messenger RNAs and a few things that messenger RNAs, depending on the type, some of them are just floating around in the cytoplasm. But some messenger RNAs can be localized to specific parts of cells, especially true in oocytes, which often store messenger RNAs long before they're actually used and long before fertilization actually occurs. Some of those messenger RNAs are going to be stored in specific parts of the cell. So, first let's take a look at the sequence of a messenger RNA. So what we're going to do is we're going to start out typical generic messenger RNA. We have our 5' cap. That's our 7-methylguanosine. As we mentioned before, that 7-methylguanosine is absolutely essential for the ribosome to recognize it as a messenger RNA, bind to it, and read it. If you don't have that cap or you have a different cap, the ribosome will ignore you. Can't bind to it. Okay, now you have a little short region, usually a few tens of nucleotides, that's not being used as instruction for a protein. We call this the 5' untranslated. In other words, ribosome doesn't read it, region. It's pretty short. And now we have the beginning of the stuff that gets translated. Our very first thing, the first what we call initiation codon, which in eukaryotes, actually it's both in eukaryotes and prokaryotes, are the nucleotides AUG. And then you have, of course, we have no more introns and exons. You have the coding sequence, in other words, the instructions, unbroken instructions to make a protein, these different codons, or sets of 3 bases. And at the very end of this, you have a stop codon. One of 3, I think it's UAA, correct me if I'm wrong, UAG and UGA. Are those the 3 stop codons? Okay. I don't always know that from memory, so okay, just want to double check. Okay, so there's 3 potential stop codons. Any one of those will lead to the ribosome not, stopping the process of translation. It's like a period at the end of the sentence. Okay, now that's all the stuff that provides the information for making a protein. But we have usually a few to several hundred nucleotides. We have what's called the 3' untranslated region. Or 3' UTR. It's frequently a few to, in some cases, several hundred nucleotides long. There's a trend in evolution that lower organisms, that's probably a pretty bad term to use, but at least organisms, less complex organisms like, say, cnidarians and flatworms and that type of stuff, they tend to have shorter regions than, say, higher vertebrates. So there seems to be a trend, the more larger, more complex an organism, the longer this region is. That's a general trend. Okay, and then finally you have the poly-A tail. And that tends to be about 200 to 300 nucleotides long. Now these untranslated regions in the poly-A tail, what they do is they bind numerous different kinds of proteins. These proteins play many different roles. Some of the proteins assist in either promoting ribosome binding and translation or inhibiting it. So you can have proteins that are translation inhibitors, they prevent translation, or proteins that are translation activators, they stimulate binding to ribosomes and start assembling the whole complex together. So we can say translation, that's protein synthesis, activators or inhibitors. We have proteins that somehow, we don't know quite how this works, but somehow protects these RNAs from nuclease attack. if you cut these sequences off in RNA, the lifespan of that RNA in the cytoplasm declines dramatically. So we'll say protection from nucleases. Protection and that regulates, of course, the lifespan of that RNA. How long does it stay in the cytoplasm before it gets chewed up? Okay, so we've got that. And another thing we have is that in some cases, proteins binding to these sequences can bind to parts of the cytoplasmic cytoskeleton. What that means is you can attach a messenger RNA to the cytoskeleton and move it to a specific part of the cell and hold it there, and maybe later on release it. So that's how you can take a messenger RNA and concentrate it into one region of a cell and not have it anywhere else. Or you can hold it to dense areas of the cytoskeleton where ribosomes simply can't get to it because it's too much of a cytoskeletal network for them to fit in. That effectively blocks translation. Or you can move a messenger RNA from one part of the cell to another as the occasion demands. So, yes?
Could you take the methyl group off of the cap to kind of regulate
translation?
after it's
Absolutely.
been in the
Absolutely. And when we take a look at regulating translation, what often happens, you hit the nail on that, what will often happen is not so much removing a pre-existing methyl group, although that could probably happen, but making a messenger RNA that has a guanosine, but not a methylate guanosine. That guanosine will allow it to get out of the nucleus. So it goes out in the nucleus, it's in the cytoplasm. But without the methyl group, the ribosome never knows it's there, so you keep it that way forever. And then when you want to translate it, there's these guanosine methylases that come up and stick the methyl group on the number 7 carbon. And now, it's all of a sudden, it pops in, it's like magic. Oh wow, there's a messenger RNA, I want to translate it, you know. And it grabs it, oh, thank you, I can translate it now. Pull it in and make a protein. Okay, but it didn't know it was there. It's like a leprechaun, just molecular leprechaun. Here's the ribosome and stuff, all of a sudden, bing, oh, 4-leaf clover and a 7-methylguanosine cap. I want it. Okay, so absolutely, and it is an important means of regulation. All right. Okay, that was actually a very good point. Good idea. So yes, you're absolutely right on. Now, as far as removing a methyl group, I'm not sure, but I wouldn't be surprised if there's an enzyme that could do that. Yes?
So this, what you've drawn is how we bind proteins. Is that how was to label that thing? What would I label it?
Oh, well, this is a mature messenger RNA. For instance, it's in the cytoplasm. And these are the important sequences. You can also bind a few proteins into this 5' region, but it's short, so you can't bind much. Most of the action is taken here and at the poly A tail. Okay, so we have all kinds of regulatory proteins here. And now, that allows us to move and localize messenger RNAs to different parts of the cell. And what does that, the best and the most dramatic, are oocytes. Because oocytes, now other cells can do that, but oocytes are the masters of it. Oocytes have a developmental program in the form of proteins and messenger RNAs laid down during oogenesis long before fertilization takes place. In fact, with many cells, if you can artificially fire off an oocyte, it will start the developmental program even in the absence of fertilization. And then it'll divide a few, sometimes even several times, and then all of a sudden realize, hey, I'm missing something. I'm missing that other genome, and then it stops. It's kind of like the old Roadrunner cartoons, you know. Roadrunner, coyote's chasing after the rotor and runs off the cliff, and he's running and he doesn't realize he's got like half a mile of empty space, but he looks like, uh-oh, I don't have a second genome. Down he goes. Okay. All right. So that's the same kind of thing. So you can actually start and activate that stored developmental program, and will continue, and then all of a sudden the oocyte realizes it needs a diploid genome and doesn't have that, and then you get developmental arrest at that point. Okay. Well, the thing is, when you look at oocytes, most messenger RNAs are indeed evenly distributed throughout the cytoplasm. But not all. Some of them have a very polarized distribution. For instance, you look at frog eggs, there are some that are extremely polarized to one, in fact, what we call the vegetal hemisphere of the cell. And that codes, that region becomes the ventral or belly part of the embryo. In fact, they have what's called VG1. Okay. And it happens to code for major signaling protein. All right, well, anyway. How does that get there? If you look at a fruit fly embryo, you find one particular messenger RNA is concentrated towards the future head. A second messenger RNA is concentrated towards the future tail. And fades out a bit further towards the center you go. How do they get there? Now here again, once again, I do want to reiterate that the majority of messenger RNAs you're going to find in cells are indeed evenly distributed in the cytoplasm. But we can take some and move them in specific locations. Now what happens though is when you look at oogenesis, let's take a look at this guy here, our protein, or VG1 is called. When you look at when these messenger RNAs are first synthesized, you find that they are indeed distributed evenly throughout the oocyte. But as oogenesis proceeds, you start finding them more and more concentrated in one region of the oocyte, and that process continues and continues, and it just gets more and more concentrated until finally it's virtually all located in that very narrow region in one part of the oocyte. Same thing with these things. So they're not like coming out of one particular region of the nucleus that's really close to their final destination. They'll be coming out everywhere and then be carried to the final destination. Now what happens here, what causes that kind of stuff? It's these proteins that bind to things like the untranslated region of the poly A tail, and then they bind to cytoskeleton and or cytoskeletal motor proteins. Now does everybody know what a motor protein is? A motor protein is a protein that binds to the cytoskeleton and then grabs onto something else and hydrolyzes ATP and walks along the cytoskeleton, dragging whatever it carries along with it. Myosin, the thing that makes the thick filaments of muscle, that's the motor protein for actin microfilaments. Microtubules have motor proteins called dyneins and kinesins. So these motor proteins, think of the cytoskeleton fiber being a railway track. Think of a motor protein being the locomotive, and think whatever that motor protein's carrying being the rest of the train. The locomotive has to use some source of energy to carry the cargo down the railroad tracks. Motor proteins use the source of energy of ATP hydrolysis, and then they can drag their cargo down. By the way, yeah, there's a big chapter in the cytoskeleton on it for review. Okay, but anyway, we find that these proteins, that these messenger RNAs, bind to the cytoskeleton. And or motor proteins. Okay, depending on, yeah.
I'm just reminding you that it's...
Uh-huh, I am just about done. Okay, and binding to, depending on what one we're dealing with, some of them bind to the microtubules, and some to the microfilaments. So we can get localized messenger RNAs and move them around. And now we can see the award ceremony. So